Hello, everyone, and welcome. Welcome to our lecture on B cells. Hopefully, last time I didn't burn you out too much with the innate immune system videos or videos rather. That was a bit much, but that's okay. You take as long as you need, or if you can, right? Today we're going to talk about B cells, which are part of the adaptive immune system. So we're moving on from innate, and of course, if you don't get these concepts, it's not the end of the world because they will come up again in future videos, right? So B cells are lymphocytes, just like NK cells and T cells. However, NK cells are part of the innate immune system, so they're very broad. Like we said, NK cells mainly look for if anything is lacking MHC, and that's like its own type of red flag or something fishy is up if something is lacking MHC. So the NK cell orders it to die. It can use like the FOS ligand, the FOS mechanism, or it could like perforate the cell with perforin and then fill it up with granzyme, which kind of like poisons it to death, right? In a sense. And then you have T cells, which operate very similar to NK cells in that regard. However, T cells are part of the adaptive. And so for something to be part of the adaptive immune system, it has to be it deals with a process regarding specific antigen recognition, right? So adaptive immune system is very specific. It's very adapted to specific type of antigens, right? Now the B cell has a misconception that it's like B is for bone marrow. It's actually for bursa fabricius, and that's it's like a organ near a bird's rear end. That's like very highly hematopoietic, and the B cells called that because B cells were first studied in this organ of birds, right? So the more you know, right? And so B cells are our body's antibody factories in a sense. If they go down that plasma cell lineage, and we did speak actually the first lecture that an antibody is basically a B cell receptor not attached to the cell surface. So the discussion of antibodies and B cells are highly intertwined. So you could have like a more correct title of this lecture is perhaps B cells and antibodies revisited, but we're not doing that. So how is a B cell receptor formed, right? Like we know that it exists and that it recognizes antigen, but what does it? How does it get there? And so it gets there through essentially a chromosome race, right? So on chromosome 14, and we have all these chromosomes, 23 from each parent, 46, right? But on chromosome 14 in specific, there's the genetic code for the light and heavy chains. And remember, because we have so many, 100 million, something like that type of B-cell receptors that we code for, there's not 100 million different genes. Rather, there's the genetic code for the light and heavy chains that are in their V, D, J, and C segments, so they're rearranged in order to mix and match to make more genes. It's a bit of a review, right? But because there's one chromosome from mom and one from dad, which genetic code is used is clearly not going to be both. So the two chromosomes race. Both start undergoing transcription, translation, DNA to RNA, RNA to protein, right? And then these proteins are made to make the B cell receptor heavy chain. And both of them will have this race, but the one that makes the heavy chain first is the chromosome that wins, right? So the mnemonic I use is 14, sounds like for the team, because it's like a race. Right, 14, right? And so again, RAG1 and RAG2 are the enzymes that direct this somatic recombination type effort. And it's the same one used for T cell receptors, the same enzyme used for T cell receptors, type of genetic mix and matching. So what's the specific order of the mix and matching? It's not particularly too important, but it could be. So again, and these lectures might not necessarily fit the scope of the course or the knowledge you're going for. Just use them as a supplement. I don't think you should base everything based on any sole source for anything in life, me included, right? Not to say I'm a thing, I'm a person, but these videos are arguably a thing. But I don't. Anyways, so the order in which the heavy chain is produced, right? First, a D and a J segment are joined together, and then they're fine-tuned, and then a V to the DJ type complex, if you will, or two gene segments, right? And then you do C to VDJ, right? And so the mnemonic I have there, C is for conclude, and then the first step to having any party, not that I do that, is getting a DJ. So that's how you know D to J is first, and then V to DJ, and then VDJ to C, right? And the C is given its name C for a reason. It's because C codes for the constant region of the B-cell receptor. And as we said, it's the constant region that determines the class, whether it's going to be IgG, IgM, IgD, E, A, or any of the subtypes like IgG3, but we're not going to talk about those right now. Anyhow, but just by default, because of the way that the genetic code is lined up, by default, a B cell will start off as an IgM and IgD displayer. Now, technically, it's M and then D, but we're not going to go too much into the discussions. IgD is not known to have much use in the same way IgM does. However, it does play some signaling roles, but there's more research going into what IgD does. However, IgM is a very famous one. Anyhow, so yeah, again, just going back, like we said, that the chromosome, right, that makes the heavy chain first is going to be the winner, and that's the one in turn that's going to provide the code for what that B cell's receptors will look like based on what they're coded for. And so essentially these chains are put up and produced and put up and then kind of like tested in a sense, like is it productive, like is it a functional protein? If it is, it will proceed then to start coding for a light chain, and if not, then it will be terminated. And mind you, only about 10% of the gene segments produced in like this whole mix and match process will actually produce a productive rearrangement for a B-cell receptor heavy chain. So only 10% of all of them were going to go on to even attempt making a light chain. And again, if the B cell can't make a productive rearrangement, it'll be sentenced to death. Now, again, if it's part of the lucky 10% who had a chromosome that could code for a B cell receptor, it will undergo the process again, but this time for a light chain. However, this time the requirement will have an additional type of step, or there's an additional requirement. There is already a heavy chain. Not only does the new light chain produced have to be functional, it also has to be able to conformationally fit into the heavy chain, because it's someone has already set the tone, if you will. The heavy chain set the tone, so the light chain has to be complementary in that regard. And so, assuming a compatible heavy and light chain is produced and both of which are functional, that B cell can be said to have a functional receptor. And mind you, there's tens of thousands of receptors on the B cell surface. Usually when you see it in the diagrams, you might see something like this usually. It's like when they give it like a nucleus and then they put up like a Y on its surface and they'll put like 2. Yeah, I'm sure you know this, but doesn't literally just have 2 receptors on its surface. No, it has tens of thousands, but for schematic sake, no one's going to draw that many. If that makes sense. Okay. And then, like I said in the first lecture, because of the genetic mix and match, and in the somatic recombination, in theory, we do have antibodies for antigens. In other words, protein sequences that may not even exist, right? So that's that essentially. So now we have the B-cell receptor, right? Successful heavy chain and light chain were produced. They indeed recognize they're conformationally apt to do their job, and they're put together and projected onto the cell surface. Now, how does this actually work? How does a B-cell receptor work? So on the tip of the antigen, see if I can. And again, this is I made the case last time that you have your crystallization region, your FC region down here, and then you have your antigen binding region here, right? And so here's just a dimer, so you could make the case that this is IgG, or maybe like 1/5 of an IgM. I don't know. So this region right here, FAB, it's a dimer. So here and here, these two parts can bind an antigen, right? And so if you were to zoom in really close on it, just schematically speaking, if you were to like to zoom, what it would look like in a sense is that it is a jumble of protein, right? And then it fits like a puzzle piece, complementary antigen, right? So it's like a puzzle piece. And it can do this twice because it's dimeric. And so again, antigen is, and for the sake of our discussion, is pretty much any protein can serve as an antigen in some regard. In some cases, I think nickel, which is a metal, can be somewhat antigenic, and then some fats and carbohydrates, but those follow completely different mechanisms from what we're discussing right now. So they will serve the scaffold of our discussion. But yeah, this is antibody here, and then again, this is antibody, this will be AB, and then this will be antigen. I'll just do A. Okay, so that's usually how it works. And so remember, we have like so many millions of these going on. So in several regards, just something to keep in mind that truly that's what a B-cell receptor is. It recognizes one specific antigen. One B cell produces antibodies, one specific type of antibody. So if you have two B cells from two different lineages, they won't recognize the same thing. If the B cell proliferates and makes clones of itself, now those clones will recognize the same antigen of the mother, of course. But that's just something to keep in mind. And then what else I want to talk about? So the B-cell receptor is a lot of drawing. The B-cell receptor, like I said, is an antibody stuck to the B cells outer membrane, right? It's truly what it is. However, it does not have an intracellular signaling component, as they're not particularly transmembrane proteins. They're more so kind of more stuck to the outside. So how does the B-cell receptor communicate to the nucleus of the cell that something was bound? It must need a signaling component, and that's exactly what it has. So if you draw like, let's just say this is the lipid bilayer, in a sense, and then you say this is the antibody, okay, and then the antibody binds its cognate antigen, okay. How does this signal to the nucleus? And the nucleus is the genetic control center of the cell. That will expose different genetic codes that can code for certain activation type proteins and whatnot. How does this communication occur? The type of it's like a co-receptor in a sense. It's not really a co-receptor. It's more of a complex. It's called Ig alpha and Ig beta. So these two are transmembrane. This is exactly what they are. They're tied or connected to the B-cell receptor, but they do have a transmembrane component as well. So upon the antibody binding an antigen, it'll trigger some conformational changes that in turn activate Ig alpha and Ig beta, and Ig beta in turn can trigger a signaling cascade that will talk to the nucleus, essentially. Now, don't get confused by the name. Ig alpha and Ig beta are not immunoglobulins that are like IgA and IgB. That's not a thing, no. Well, IgA is the thing, but it has nothing to do with Ig alpha. This is just the name of this complex that assists the B-cell receptor in signaling. Okay. So just keep that in mind. So cross-linking is an interesting type of thing, and I think one way you could probably say you have the B-cell receptor, right? You have the nucleus, Then you have your Ig alpha and beta. Okay. Okay, we'll just say this nucleus. And then you have your cognate antigen, right? This individual antibody is going to bind the antigen and have its Ig beta signal to the nucleus that, hey, I found something. Now, if we have several thousand antibodies or B-cell receptors technically on the surface of the B-cell, doesn't it seem a bit odd that only one or only one B-cell receptor is strong enough amidst the crowd of 1,000 to make a noise that's hearable by the nucleus? It really isn't. Therefore, we need to have something called cross-linking. And so basically what cross-linking is that when the several B-cell receptors that bind antigen actually kind of aggregate near each other, and they kind of make this region on the cell surface that's very, full of cross-linked antibodies. And together, through the power of collective force, they make a loud signal. And this loud signal, in turn, is actually what can kind of trigger activation, right? So one voice isn't enough. You need so many voices, and you need these voices to come from cross-linked B-cell receptors. And so I don't want you to think of it weird as that the cell membrane is some fixed type of thing. Remember, it's a fluid mosaic, so proteins inside of it can like shift around. You know what I'm saying? And so that's what happens. They shift around, and usually they shift around in a region where the B cells like facing the pathogen or whatever source of antigen there is, right? And so this is very important. Now, this will produce an activation in something called T-cell independent activation, where enough cross-linking happens that a co-stimulatory signal or a helping signal from a T-cell is not required. And that's an important concept I introduced now. In order for B-cells to be activated, typically, not only do they need cross-linking, right, but they also need T-cell help, right? So T-cells help B-cells in order for activation. However, if cross-linking happens to a great enough extent, it can actually have a signal so strong enough that it overcomes the need to have T-cell help. Now, B-cells produced without T-cell help are kind of dumb in that they're not as sophisticated, in that they're not as capable as T-cell assisted B-cells in having more specific progeny or developmental paths, rather, not to sound too pretentious. So just be cognizant of this type of thing, so that T-cell independent activation can happen if cross-linking happens to a great enough extent. We're going to talk more about this later. I believe it's B cells in the spleen that are pretty good at doing this, but I need to confirm. But we'll talk more about this in a later chapter. But that's cross-linking. Whether you're T-cell independent or T-cell dependent, you need to have cross-linking in order to have an activation signal. But in the case of the T-cell independent activation, a great enough amount of cross-linking can actually overcome that requirement for T-cell help. Okay, so the complement co-receptor is another surface marker or on the surface of the B cell. So right now we already established that the B cell has the B-cell receptor flanked by Ig alpha and Ig beta to assist in intracellular signaling, but there's also complement receptors, and specifically it's for IC3B, and this is a more dormant form of the C3b that is produced from either a spontaneous degeneration of C3 into C3a and C3b, C3a being an aflatoxin, C3b depositing on the membrane of a pathogen, or it could have been produced by a C3 convertase, either C3BBB or C4AC2B, right? C4BC2A. Sorry, I had a brain fart there. C4BC2A, which is the MBL or the classical C3 convertase, and the other one being the alternative, right? Anyhow, so you have C3 deposits on the membrane actually go into this more dormant form called IC3B, which is a opsinin, as we established. And so this complement co-receptor can signal to the nucleus essentially, oh, hey, the fact that there is IC3B depositing on this membrane means that our innate immune system has already tagged this specific surface as being that of a pathogen. Therefore, we know it's already dangerous. Therefore, to trigger intracellular signaling and activation, the B-cell receptors don't have to have as high of a threshold for cross-linking in order to activate the B-cell, because the whole point of the cross-linking is more like an "are you sure" signal. But the fact that there's IC3B deposit on the pathogenic surface means that the innate immune system has already confirmed that there's something dangerous here. Therefore, there's no reason to ask, "Are you sure?" a third time. At that point, you're just gaslighting, right? That's one way to put it, right? So again, the complement co-receptor makes the B cell more sensitive to antigen and lower the threshold for cross-linking, because again, the fact that IC3B can even deposit on the surface mean that, hey, it's already something fishy. Because mind you, we discussed that our body has its own physiological mechanisms of making sure C3B doesn't deposit on our own cells, right? Right. This is a mental and visual break. Breaks over. I'm sure you could have paused the video. Actually, if you want, you can pause the video here. I'll smile for you. Okay, now we unpause. So we spoke about the T cell independent activation. Now I'm going to tell you about the T cell dependent B cell activation, and I kind of summarized it for you already, but we get more into a little bit more specifics. So again, the T cell independent B cell activation can occur if there's a lot of antigen, right? But in some cases, there won't be a lot of antigen present. So the antigen could be a red flag for something quite dangerous. So activation could still be necessary. So what does the B cell do? Stop its feet and say, no, I demand a lot of antigen? No, it doesn't. It becomes a little more flexible. It has mechanisms in place to ensure that even if there's not a lot of antigen present, it can still activate. And this is through T cell help. And so the B cell has a surface marker called CD40. And again, the CDs are just cluster differentiation. It's a biomedical term. You don't have to worry too much about how they name them and whatnot. So this is the B cell, right? It has something called CD40, and this is what this is going to be, right? And the way I like to remember it is that B for B cell reminds me of like a ball, right? And then when I do like oh, very good, not good handwriting. When I think of a ball, I think of like, a circle, right? And then I think of like a B cell as a ball, and then I think about the 0 and 40 as also a ball. Whatever works for you. But CD40, because CD40 really is a B cell thing, right? So you won't really see CD40 too much in the context of other types of cells. You could make the case, though, that I won't get into it too much, actually. Maybe later. So CD40, you can think of CD40 as a button, and the helper T cell has, has a ligand that can essentially lock into CD40. It's called CD. So ligand has a corresponding protein to it, right? So CD40 ligand binds to CD40. Pretty self-explanatory. So CD40 is on the B cell, CD40 ligand is on the helper T cell. When this happens, a co-stimulatory signal is given, and CD40 does have an intracellular type mechanism as well. So when the nucleus receives two signals, the signal from the B-cell receptors, Ig alpha, Ig beta, with all the cross-linking to the nucleus, it gets that signal that, hey, there's an antigen, let's activate. And then it gets the co-stimulatory signal from the T cell via CD40L binding to CD40 on the B cell. It says, okay, yeah, the signal is all clear. Once you have this co-stimulatory signal, the B cell can indeed activate. Okay. So this is just pretty much a repeat of what we're saying. For it to be an independent activation, if there's just so much cross-linking happening, it can kind of overcome the need for a co-stimulatory signal. But mind you, even for this to occur, the B cell must know that there is a fight that is even on. Otherwise, we'd have an autoimmune mess. So how does the B cell know that there's a fight? How a fight is on? There's a couple ways. It could perhaps detect on some cytokine that are only present in inflammatory situations, or even some B cells have toll-like receptors, much like macrophages and natural killer cells, that have evolutionarily kind of picked up that okay, hey, if this damage-associated molecular pattern is present, that we know that there's something wrong. Picked that up. We picked that up evolutionarily. So either of two ways, but as long as the B cell knows something dangerous is going on, it has an inclination to allow the cross-linking to proceed in activating the B cell, right? And again, T cell independent activation is useful. It's useful in several regards because to produce a T cell helper T cells are also part of the adaptive immune system. So that means because they have to undergo a process of antigen recognition and development and spreading and all these things and aren't just readily present, just like macrophages and whatnot, like the innate immune system. T cells and B cells like take some time to develop, right? Especially if it's the first infection. Therefore, awaiting for T cell help can take some time. So early on in infection, some T cell independent activation, because there's no T cells, can actually be useful for some B cells. And again, we'll talk about marginal zone B cells that are a special class of B cell and the lymphoid organs that just operate normally without T cell help. So the B cell is activated. Now what? We've made the B cell. We tacked it on the surface. And recognize an antigen, use their Ig alpha and beta and our co-stimulation from CD40 binding CD40L on the T cell receptor, or if it's independent, then you didn't need that, and to send a signal to the nucleus, and the B cell activates. Now what? So certain things are now produced into proteins and upregulated within the B cell. And so we're going to talk about the specifics of this, because this is not specific at all, but we'll talk about the specifics of this in a later lecture. But more or less, now that the B cell is activated, it's turned on, it can actually do more things, right? So one of the things it can do is class switch. So it starts off as IgM and IgD, recall, but now it can actually class switch to become another class. And that's important because let's just say you have, and we spoke about, I believe, some of the classes before, and that we can go more into them. But let's just say you have an epithelial type infection. You're not really going to need IgM. You're going to need IgA, right? So, but if you're by default only producing IgM, it's not good. So the activation helps because it can get you more specific isotypes of the antibodies you need, and also the B cell can undergo somatic hypermutation. And that's basically when the kind of the tips, the antigen binding region of the antibody is fine-tuned so that it sticks to antigen better. It's like a fine-tuning process. So hypermutates a bit so it can become fine-tuning. And then the B cell will make a decision if it's going to become a plasma cell or a memory B cell. A plasma cell being an antibody factory, a memory B cell being a more dormant type cell that if it recognizes cognate antigen after the infection is done in, like, say, a second infection, it will readily proliferate into a lineage of fast-acting and highly adapted B cells. It's like that's what hence the name memory cell. We talk more about specifics on this in a later lecture. So again, to discuss the class switching, just again recall that the FC region, the constant region, the crystallizable portion, right? The genetic code there determines the immunoglobulin class. So like I said, IgM and IgD are the two. So the FC region genetic code is edited. So the C region is usually what's messed with in a sense. Okay, and we'll talk in a later lecture about how the B cell knows that, say, the antigen is located in the epithelium. It has a lot to do with the cells that present antigen or carry antigen, but we'll talk about that later. But yeah, antibody isotype is a very important concept. So here, I guess we do the formal introduction, and we've kind of gone into these before, so hopefully it can help a little bit. IgM is a pentamer. So as we discussed, I believe it was in the very first lecture, that means it has 10 binding sites. Again, it looked like the snowflake. So if you're doing like holiday cards now and you want to be like very biomedical or medical or something about it, you can do like blue um, uh, IgMs on your cards that look like snowflakes. I don't know. And it's the first type of antibody produced by the cells in the immune response. It can fix complement. So you have to recall that C1q in the classical pathway cannot become proteolytic against C4 and C2 unless C1q has scaffolded or docked onto two IgGs or one IgM, right? And because the IgM is so big, 10 binding sites, it almost has like 10 arms, if you will. That means it can grab onto a virus or something in 10 different places, like hugging it, and that really debilitates it. So it's a very good neutralizing type of antibody. As opposed to IgG, that's a monomer, so it looks just like 1/5 of what we drew above. Right, this looks like a Y. It's the only immunoglobulin that can cross the placenta. So that's how what one means of passive immunity, in which the mother confers some immunity onto her baby. And you can think of this way, remember it, because IgG is only a monomer. It's quite small, and therefore it's more easily crossable across the placental barrier. That being said, I think there could be some problems with IgG being able to cross the placenta if there is some incompatibility between the mother and the fetus. It's for this reason that hemolytic disease of the newborn is a problem. Fortunately, because of the advent of ROGAM, it's not as much of an issue today, but usually if there's blood type incompatibility between the mother and the child, on the mother's second pregnancy, usually, that child could actually be targeted by the mother's IgG molecules, tagging a lot of the developing child as a foreign and dangerous substance to be attacked. And the children could actually be killed, or the fetus rather, or child, I guess, yes, can be killed or have a lot of other damage if they aren't. They'll have a lot of anemic problems. They'll be born jaundiced. In the olden days, and I still think they do it now, one way to treat the jaundice is to put the newborn under a UV light, right? But this is a big tangent, sorry. Aside, we can maybe have our own lecture on hemolytic disease of the newborn. Anyhow, a big thing you should know about IgG is also that they're the antibody that mediates something called ADCC, and you're going to hear ADCC a lot. They like it for some reason. It's antibody-dependent cellular cytotoxicity, in which IgG tags cell for destruction by an NK cell, in some cases also a T cell. But in this context, we say NK cell. So basically, if a cell, right, has IgG stuck to it, it can help that cell get opsonized by a macrophage or something, or get tagged for destruction. And that specific tagging for destruction can be mediated by NK cell, in which case we call it ADCC. And so again, the NK cell can recognize something as dangerous because it's missing its MHC class 1, but also it can do it through toll-like receptors, or it can do it through ADCC, in which if it's basically deduces, oh, hey, if a ton of antibodies, IgG antibodies are stuck to the thing, it must be dangerous. Therefore, I think it's safe to say that I can go ahead and destroy it. IgG is actually the most abundant immunoglobulin in the blood, but not most abundant in the body. The most abundant in the body is actually A, and that's a trick question that some professors like to do. So just something to keep an eye for. IgA is the most abundant immunoglobulin in the body. It's on mucosal surfaces, and when you consider that our, I think, large intestine unrolled is like bigger than a tennis court, I think, because it's highly wrinkled to maximize surface area. So it makes sense that IgA is the most quantitatively numerous. Usually it's dimeric, and that might sound a little bit funny, but maybe it is, I don't know. Usually the way that works, that it looks something like this. and then there's usually like a protein clip tethering the two. And actually, this protein clip is very important because this protein clip helps this specific class of antibody to be able to move between endothelial cells and epithelial cells rather, to get from place to place. Unfortunately, some bacteria have adapted to being able to recognize this clip and being able to cleave it to render IgA less effective. But it's a very interesting little curious protein fragment that's worth knowing about, and it also holds together the two monomers to make a dimeric. And also it confers some acid resistance onto IgA, and as you can imagine, the GI system is very acidic because of the stomach, so it's very good to have some acid resistant proteins, immunoglobulins, right? IgA is also found in the mother's milk, which is another form of passive immunity aside from the IgG coming through the placenta, and they work along mucins to protect the mucosal surface. And the mucin is just basically a giant carbon compound. It's like one of the larger molecules we have in our body. It's this big log of carbon that's like really good at like catching things in a sense, right? So mucins and IgA are quite abundant in mucosal surfaces. And then also you have something called IgE. This is very famous and very important. And when we talk later about Th1, Th2, mast cells, and all these other type of things, it becomes more pertinent to discussion. But if an allergen, which is a specific type of antigen that triggers an allergy, is recognized by a B cell, that B cell will become activated, class switch, whatnot, and it will switch from IgM and D over to IgE. And then this IgE antibody produced by this B cells will dock on the surface of the mast cell. We didn't talk about mast cells yet, but they're one of the resident cells that we have in like surface of skin. So let's just say this is a mast cell, right? So on the mast cell surface, it actually has receptors for Fc regions of IgE antibodies. And so then here comes an antibody. And so you're used to let's just say this is an antigen. You're used to antibodies binding something like this. So the FAB regions and FC regions just like hanging out to assist in opsonization by macrophage or something. And the antigen binding regions are here. But this receptor here actually does not bind to the mast cell the same way it binds to a pathogen. Rather, it docks onto it. So the FC region is what binds to the mast cell, and then the antigen binding regions are out here, right? And so now basically you have a mast cell holding the IgE, and this antigen grabbing hands are essentially out. So in a sense, you could say that the mast cell like extended its receptor, right? And so mind you, this antibody is still specific to the allergen, right? Because it was produced on the B cell, and that B cell recognized the allergen, and so because everything that comes out of the B cell recognized the same thing, it just recognized the same allergen again. But essentially, I call this position locked and loaded. So you have the mast cell that has the IgE antibody. If that allergen is encountered again, and it's encountered by the mast cell, the receptor on the mast cell will actually detect that the antibody has bound something, and in turn will signal the mast cell to do something called degranulation. And this is significant because the mast cell is full of granules of highly vasoactive compounds, and basophils and mast cells are, because they have these granules, they're very prominent in allergic responses. And the mnemonic is like IgE allergy, and I think I have that down here. Also parasites as well, but that's aside the point. So after being locked and loaded, and then the IgE tethered to the mast cell surface recognizes the antigen, the mast cell will degranulate and release all these vasoactive substances like histamine and whatnot. Okay. Yeah, and the technical term for allergic reaction is anaphylaxis. It's not to be confused with prophylaxis, which is kind of like in pharmacology when you kind of begin a dosage of a drug as a preventative measure. Side of the point. Okay, and so how does a B cell know to become an IgA producer, an IgG producer, stay as an IgM producer, or whatnot? It's usually subject to the isotype. It's subject to the cytokine environment in which the B cell is located, and so the helper T cells secrete these chemicals, and these chemicals are basically a recipe, right? And it's kind of like this. So, like hot pot. It's a very delicious experience that I highly recommend. If you're cooking the meat in a tom yum broth, right, you're gonna get tom yum flavored meat. If you're cooking it in a Szechuan broth, you're gonna get a Szechuan spice type flavored meat, right? Um, and that's exactly what this is. So the B cell gets cooked in like a broth that kind of determines what flavor it will be, right? And so if the broth is IL-4 and IL-5, and the broth producer, if you will, is T helper cell, you're going to IgE if it's interferon gamma, which is, mind you, the same molecule that wakes up macrophages from their relatively dormant phases, dormant type thing. Yeah, but it also promotes class switching of B cell to Ig3. And so Ig3, as you can imagine, is very involved in macrophage activity. So it's actually very cool that the interferon gamma, not only does it wake up the macrophage, it also makes the B cells more adept at producing antibodies that the macrophage is better at detecting. And a macrophage is very good at detecting IgG3. Okay. And again, the way it happens is like the macrophage is very good at picking up, detecting the FC region, use it as an opsonin. He knows, okay, well, we have all these opsonins on this pathogen, therefore I should eat it. Exactly. TGF beta promotes the class switching to IgA. TGF beta, we'll talk about it later. It has a very important roles. It's called transformation growth factor. In modulating the immune system, and then also the GI immune system, which is actually a little bit of an independent entity relative to the systemic system that we have going on. But we talk about that later. And again, how does we take we take it one step further. So if the B cell knows how to class switch based on the cytokine environment, how does the T cell producing the cytokine environment know what that's like, which way to direct the B cell? And that's what I was saying before. Specific antigens know how to direct the B cell, but also the antigen presenting cells to the T cells can also direct the response, and they can confer they make like a dossier of information. They're like, hey, by the way, this antigen, it was found here, and it's most likely gonna call for you to make these type of cytokines to trigger these type of cells and trigger this type of isotype switching. It's a very sophisticated process, not fully understood, but we'll have a whole lecture on it at a later day. And so now we continue. We have part of me feels like I should have split this lecture into different parts, but we'll finish. And even you guys take breaks. You take the pause here. I can smile again. Okay, unpause. Now you can. Welcome back. Okay, somatic hypermutation. I already discussed it. It's a fine-tuning process after the B cells activate, in which it fine-tunes its antibody to have more stickiness to antigen, right? So basically make the antigen better at its job, right? And this is actually facilitates something called affinity maturation. It's a very Darwinian type system. It's a very survival of the fittest type system, in which the only B cells with receptors with strong enough affinity are the ones that are like most likely going to have the all clear to produce a good enough signal to fully mature and go meet a T cell that will give them co-stimulation so they can make a progeny, right? So it's truly like only the B cells who are best at binding antigens will have the opportunity to fully mature with the help of a T cell, in order to make a progeny, to have descendants, if you will. Okay, we'll talk about this later in lecture. Okay, and we discussed this already. Difference between a plasma and a memory cell, and this is the key point I wanted to make down here, though. Memory B cells cannot be produced from B cells activated by T cell independent activation. And this is believed to be because the CD40 on the B cell to the CD40L on the T cell interaction is believed to confer the biomolecular requirement needed to give that B cell the ability to, you know, produce, become part of the memory lineage, right? So usually we can think of the ones that are the B cells produced by T cell independent activation as very short-term type use. They won't form any memory, but they're needed in the moment, and that's okay because we don't have T cells available yet. We'll take what we can. As opposed to these memory B cells, they do have memory, but in order for a cell that can remember stuff to be made, some more work has to go in, and that more work is going to be the CD40 to CD40L interaction. Okay. Wow, that was a lot. Well, I thank you all for tuning in today. I hope that was helpful. Next time we'll talk about antigen presentation, and this, of course, you can imagine is going to deal a lot with T cells. And I think the next couple lectures are going to be mainly T cell heavy. All the concepts brought up today will be brought up more later and more specifics. So if you didn't get it this time, I'm sure you can rewatch the video, but just keep in mind that we will be building on this in the future. And these are my sources. Thank you for tuning in again. And I wish you all well. Take care.